All right. So for Jasmine, who's joining us for the first time, we just each person takes a figure or volunteer to take a figure, and then we just go through the paper. And it's very informal. So if you have any questions, just ask. Doesn't matter what the question is. Right? Don't be shy about asking questions. Right? The goal of the journal club is to understand the paper. Right, so let's see. So I think the paper follows the theme that we've been following that we've been taking for the past couple of weeks, where we're looking at manipulation of the entire. So we're on what, paper number 31 or something. This entire series has been mostly about feeding circuits. We've thrown in sprinkles of the taste system and some other thermal regulation, but mostly we focus on feeding circuits. And when people talk about feeding circuits and neural control of feeding, the most basic brain region that does this is in the hypothalamus, in the arcuate nucleus of the hypothalamus, and in particular, these neurons that express AGRP. So there's a mouse strain that expresses Cre in the AGRP neurons, and so this paper makes use of that mouse strain to see so these essentially are hunger neurons. So when they're active, the animal feels hungry and they'll consume more food. So what they explore in this paper is what happens when you stimulate these hunger neurons for a long time. So I think it's a pretty simple paper, and we'll get started with figure 1. So is this too big? Should I zoom out a little bit? No?
I think
Good.
it's good.
Amanda, you're muted.
Hello.
Hello.
Yeah, no, this is fine. And then when I get to E, you can just scroll down.
Okay.
Surprise, I took figure 1 as the first one, so I grabbed it. So basically this paper, they want to see if chronic excitation of the AGRP is enough to lead to obesity. And so they wanted to look at the acute effects first. So they activated the AGRP neurons with GQ coupled receptors. They transduced AGRP Cre mice with Cre dependent GQ coupled designer receptor and just M cherry as the control. So that's 1A. Figure 1B is just a histology of the AGRP neurons in the arcuate nucleus. So what they did was they injected them with CNO and measured their acute food intake. And so the one with the GQ receptor ate much more than the wild type mice, and this was statistically significant as seen in figure 1C. But figure D shows that after about 4 days, the mouse's eating level returned to baseline. Also, something they found that was interesting was that the mice with the receptor, they were pair-fed, meaning that the food quantity matched the controlled mouse's food intake, but they didn't finish their food. So maybe their appetite actually ended up decreasing. These experimental mice gained weight initially and then lost it. And then the little inset shows that when they removed chemogenetic stimulation, the signaling pathway was still working. That's the one part I didn't really fully understand. But then figure 1E shows that the relative body weight, and that by day 59, all of the mice ended up with a similar body weight at the end. The end.
Very good. So the inset is they're comparing the amount of food eaten after 63 days. Wait, actually that's not true. So here they present CNO, right? for how long?
Yeah.
62 days, right?
Mm-hmm.
And then
Yes.
they take it off for 1 day and then stimulate again, right?
Yeah.
That's the days to 63. Is that right?
Yes, I believe so.
Yeah.
I looked it up, but I wasn't exactly sure what the CNO does.
Oh,
Like I looked up what it is, but I wasn't really sure like what the possible, because they keep going with the CNO versus non-CNO, and I just wasn't able to figure out like what exactly it does.
okay, yeah. So this this M3DQ receptor is a receptor that will bind to CNO and activate the neurons. It's the easiest way to put it. So the ligand for this synthetic receptor is CNO. Is that making sense? So this DREAD experiment, which is a way of activating neuronal subsets. So let's just look at the experimental setup here. So this animal that has Cre expressing in the AGRP neurons, right? So the Cre is in the AGRP locus. So in any cell that expresses AGRP, these cells will express Cre. And in these animals, what they're doing is they're using stereotaxic coordinates to inject viruses. In a subset of animals, they're injecting this AV1 DIO DQ mCherry virus, or in the control animals, they're using DIO mCherry virus. This DIO stands for, what does it stand for? Double inverse orientation. I think that's what it means. But essentially, what it means is that it takes Cre, and when there's Cre here, then the DQ protein that's there, or the construct, will now be in the correct orientation, so that it can make the DQ. In the absence of DQ, in the absence of Cre, it'll be in the inverse orientation, so it won't make a functional DQ protein. Right? So in the AGRP neurons, because there's the Cre, these neurons will now express the DQ protein. Right? In the animals where the DQ was injected. Yeah. Making sense so far? I'm looking at you, Jasmine. I think everyone else has been going through this, and I'm trying to make it simplify this, right? And the reason for using viruses as opposed to a different mouse line, first of all, is that you can target the DQ to be expressed in the brain region that you're interested in. You can also do this, start this expression in adults. So if AGRP is expressed earlier on during development, then it doesn't get expressed in other areas that you're not interested in. So this Cre is a recombinase. It's an enzyme that will take a target sequence called LOX, and it targets the LOX sequence and then recombines. So in this case, what the recombination, what it'll do is it'll flip the orientation of the DNA. So without the Cre, it's in the antisense, so it won't make a functional protein. But when Cre is there, it'll flip around to the correct orientation and it'll start making the DQ. Making sense?
Yeah, it makes sense.
Okay. The DQ also is tagged with a red fluorescent protein called M-cherry, so that we can see. I don't know if Amanda, do you know if B is an image of the, what is this image of?
It was showing like food intake, but more acutely is where they were looking right here at how they eat a lot more, and then they eventually return to baseline.
Panel B.
Oh, B.
Yeah.
It said it's a histology, and so then I looked more specifically at the paragraph where they cite B, and it says it was a little unclear, but I think it's the arcuate nucleus.
Yeah, I'm just wondering what the fluorescence
I'm
is.
not sure what the point
It
is.
could be, say, any of the Dio-M cherry or the DQ-M cherry, but they just want to show that they're targeting the arcuate nucleus.
Yeah, I think so.
I have a question.
Mm-hmm.
So, are there any endogenous neurons that could potentially, because this is chronic activation, they're giving it for 10 days, however many days. Are there any endogenous neurons that would be affected by this? Like, I know the DQ receptor is specifically like binds CNO, but are there any endogenous neurons that have receptors that could also bind it that like could affect the results that we're seeing?
You mean in the absence of the DQ?
In the absence of DQ, like do mouse neurons in general, any neurons, have a receptor that, you know, either could bind CNO or a metabolite of CNO? Like, this is present every day, and I'm sure it's been broken down by enzymes. So I'm just wondering what are the targets, like the effects of that are.
So there's been a lot of debate on the side effects of CNO. So presumably CNO is supposed to bind to the DQ, and it works specifically through this receptor so that you only activate the neurons of interest. But as with many things, if you have CNO floating around in your body for 60 some days and at pretty high concentrations, who knows what that can do, right? Jahao, do you remember any of the more specific side effects of CNO?
I think not directly from CNO, but from its metabolism, like clozapine.
Mm-hmm.
Clozapine is one of the metabolic, like downstream. I probably not remembering the specific terms for that. So the CNO can be, so the CNO can't cross the blood-brain barriers. So it has to be first metabolized in the kidney by an enzyme or a set of enzymes. And clozapine CLZ is one of the metabolism products of those processes. And so even though CNO has been shown like the in vitro, so without any kidney processing, can bind into the drug, but in vivo, I think the most potent they say components of CNO effects is from clozapine. Mm-hmm. And one of the effects, clozapine has been, I think, is used in the psychiatric applications. So there are some side effects, probably due to, probably through the like MNDA or AMPA receptor. So that side. I'm not exactly sure, so I have to check back.
Amanda, do you have anything to add since you looked up Wikipedia for CNO?
No, I saw it was like a related to dreads, so
okay.
I kind of narrowed down what it was.
Yeah.
But specific to Nancy's question, there is M-cherry control here.
Yeah.
So specific to their assay, that's all. No, they can do.
So that's a good point, because when you do experiments, you want to do very good control experiments also, right? So in this case, they could have compared the the DQ injected animals, right, with or without CNO, and then they could have compared those groups, right? But here, what they chose to do is to compare animals that have the DQ versus animals that don't have the DQ. They went through all the same procedure. They had a hole drilled in their skull. They had a glass needle go in and deep into the brain, right? It's almost at the bottom of the brain. Go deep into the brain, inject 200 nanoliters or whatever volume, quite a large volume of liquid containing viruses, right? And then all of these mice got CNO. So if there is a side effect of CNO that makes animals eat more food, then you would have seen it in both the M-cherry and the DQ conditions. But here you can clearly see that it works much better, or it only causes food intake to increase in DQ groups. So I think we all know that even when we are hungry, right, we have that phase when we feel really hungry, and then maybe it'll go away, and maybe it doesn't last for 4 days, right? And that's exactly what you're seeing, that the body is making adjustments so that initially, when the hunger neurons, the AGRP neurons are stimulated, the animal thinks that it has to increase food intake because it's hungry, and it'll do that for a couple of days, but after a while come back to the normal or the regular food intake amounts. And I think this, and it's a pretty simple paper, but it speaks to the power of these tools that we have right now. The ability to, so the AGRP neurons have been known for a long time to be involved in feeding behaviors, because people who have hyperplasia or cancer and to where the arcuate nucleus AGRP neurons are more activated, then they'll feel more hunger, and they might increase. They might have increased obesity, prevalences in obesity, or when there are lesions or you suffer a stroke and that area is damaged, then they might feel like less hungry, and they might not eat as much as they should. And so in the old days, when people were doing experiments in mice, these were the kind of experiments that people were doing. They were taking out the AGRP neurons or the arcuate nucleus and seeing the effect of a lesion in the brain of mice, right? Or they might stimulate, putting in electrodes and stimulate that region and see what the effects are. But now you can do more controlled experiments by targeting viruses in a mouse strain to target just the AGRP neurons, and then use this DREAD technology. DREAD stands for Designer Receptor Activated by Designer Drugs, meaning that this receptor, the DQ receptor, is activated by CNO. So you can do these kind of long-term experiments where you stimulate a set of neurons for very prolonged times. And I think that's why when we did when Jiahao did the DREAD experiment to stimulate the taste system, we see things that we weren't expecting, because this is such a strong stimulus. Okay, okay, so I think we can move on to figure 2. Right? So who Who will go over figure 2? Nobody wants to do figure 2?
I have another question. Sorry, I'm just still on the clozapine versus CNO. I just realized that they administered to the water either or.
Mhm.
And I'm just like, why?
Why water?
No, why either? Like they're different molecules, and like clozapine, which is what they apparently might have added to the water, also has different targets. So that's why I'm wondering, why not strictly CNO? Why do they go back and forth between clozapine and CNO?
Yeah, I didn't catch the question. So you're saying why did they?
Yeah, why did they administer either clozapine or CNO when I thought CNO was the ligand of choice for DRESS? Why would they add clozapine instead?
So
Right? That's what this experiment and the previous one, it's like they added clozapine to the water.
mhm. So how did they add this?
In their mixers, they have, like say, 25 microgram per meals for CNO and 1/10 of that for clozapine. They
Mhm.
first dissolve them in DMSO and then put them. So once the chemical is dissolved in DMSO, then the whole thing can be readily dissolved in the water. So it's kind of like a mixture. That's how they do it. So I don't know exactly why they use both of these, DREAD agonists.
Mhm.
Actually, people either use CNO or clozapine. Clozapine is much more expensive than CNO. And on the other hand, clozapine is not the only metabolism coming from CNO when CNO is metabolized in the body. So it could be that they want to see, try to kind of see like whether it's exactly because of side effects of CNO, whatever. But exactly what, I don't know. Based on their figure 1, it seems like they pull the data for CNO and clozapine group. Then they didn't differentiate them in their figures.
Yeah, I don't know if there's a big difference between reported effects of CNO and clozapine.
Yeah. But clozapine is a drug though. So I think I recheck it is targeting the serotonin receptor. So basically dealing with depression, anxiety disorder, something like that.
Mm.
So it could be, more like clinical relevance if there is like a commercial or available drug structurally closer to clozapine versus the CNO. Not sure.
That's a good point.
Some of those, like said, yeah, psychiatric drugs have side effects on the body weight, feeding, something. So could be relates to that.
So Jahao, you want to go over this figure since you're probably the only one who's done some slice recordings?
I know, but okay.
Yeah.
So from first figure, when they take off, from the water at day 63, they still, so you still see a drop of the food intake. So basically that means when CNO activation to the AGRP neuron is not there, then the animal loses a little bit of drive to eat. And then they're wondering, to further confirm this, just try to see directly how CNO affects the neurons. So the first thing they do is they slice the brain. I mean, the neuron's still alive. So there is a technology called I'm not sure exactly like this, but they do the brain slice, put them into the ACSF. It is artificial cerebral spinal fluid. So it's an artificial environment that enables the neurons to fire in a relatively close to a physiologic environment. So the neurons normally can live up to, let's say, 6 hours to 10 hours. And then during these environments, they can change the components of ACSF. So here they add the components CNO into this ACSF. And what they shows is in the control slice where there's like first account, so those cells have never been injected with CNO before. So the CNO effects there as expected with increase of spike firing. So as the vertical line is a spiking. So each vertical line is one spike from actual potential. And then after like the pronounced CNO intake, the effects is still there, maybe even more dramatic. And so they quantifies what happens in their recording.
Question. Not for Jiao. Jiao probably knows. But, so how many cells are in this panel A? Let's take this guy here. Whose activity are they measuring? How many neurons?
Isn't this just spikes from a single neuron?
Yes. Who said that, by the way? Oh, was it Nancy?
Yeah.
Nancy, and I was also holding one finger up.
Okay.
I have a, well, no, we haven't gotten there yet. Sorry. I'll ask my question when we get there.
Okay, so then if it's a single neuron, which neuron is this?
Like in the control ACSF?
Yeah.
It's one of those M-Cherry.
Oh, good. So how would they find that neuron? You've already said it, but right? So Nancy said it, but Nancy, you gave away the answer.
Sorry.
But they're targeting the AGRP neurons because the AGRP neurons have fluorescence, right? So when they take the slice of this brain and put it under the scope, they'll be able to see neurons that are labeled with red fluorescent protein. So then they can go in and patch those neurons and do electrophysiology specifically in the AGRP neurons, and that's what they're doing here. Right? So this is a representative trace of an AGRP neuron in the arcuate nucleus, and they're looking at activity, spiking activity in these neurons. Right? And you can see that probably this mouse never saw clozapine. So this is what the activity looks like, and then you put CNO, the AGRP neurons fire. This is a mouse that have been treated with clozapine for 10 days. You can see that there are less spikes than the control animals, right? So they're less active. But when they put CNO, so CNO in this case will be flowing through in the solution that the brain slice is bathed in, right? And you can see that, but upon CNO administration, you see a lot of spikes, and that is tabulated in the graphs below, which Jiahao will go over. Yeah. Yeah.
So B and C is the statistic comparison between the let's say naive case versus pronounced clozapine drinking case. Mm-hmm. Baseline, there is a decrease in the firing of AGRP neurons, And then upon signal activation, they reached almost the same level of activity. So that means the dreads, so the signal effects on dreads are there, even upon the chronic. The one of the reason, not the reason. So one of the consideration is dread is even though it's an artificial receptor, it's still based on the intrinsic GPCR receptor. The GPCR receptor has some, let's say, desensitized mechanism. So upon prolonged stimulation, the receptor can, let's say, I think it's not trafficking. The receptor, let's say, gets down from membrane to the plasma for recycling, probably for degradation, for other things. So that will reduce, that's presumably will reduce the potency of the receptor activation. So in their case, pronounced activation of dread doesn't affect this potency. And then in D, so the D is another way to comparing the effects of the CNO. And it seems that they are, so even with a pronounced clozapine intake, it's probably like the potency of the dread seems greater. And then in E, they're trying to say since there is observed a baseline decrease of firing rates, so what happens to those neurons? Are they receiving, let's say, less excitatory inputs, or are they receiving more inhibitory inputs? They look at those by different modes of, so this here is the, each spike is indicating a current. Depends on the orientation of the spike. On E, you have the, let's say, upward spike, and in H, you have inward spike. So this indicates, in E, it indicates the current that flow out of the cell, right? Yes, it flow out of the cell. And so that would be indicating the IPSC. And then H is the inward current. So current that gets into this from the outside of the cell and into the cell. So the effects of those current is to excite the neurons. So if you look at EFG, that means with 10 days of clozapine, the neurons get more inhibitory inputs, so the spontaneous iPSC. That's well, I need to remember what that I mean. Oh, inhibitory postsynaptic current.
Yeah.
And then that gets a little bit larger, a little bit like twofold, but the frequency, so the frequency increase, but amplitude doesn't. So that means the effects are coming from the presynaptic. So there's not theory, but there is a common way to differentiate. So the differentiate the synaptic effects, whether it's coming from the presynaptic or postsynaptic. Usually the frequency change coming from the presynaptic and amplitude coming from the postsynaptic at the receptor sites. So that means the presynaptic inhibitory input to the AGIP neurons increase upon the chronic clozapine application. And they draw another parallel conclusion on the excitatory based on the HIJ. Basically, there's no observable difference or effects on the excitatory part. Yes, that's it.
I have many questions.
Okay.
Okay, so first of all, I have questions on B and C. what would the normal frequency be of of just a regular neuron? Because a frequency of 5 hertz seems very low to me, but I'm not sure if this is just because these are AGRP neurons or if this is like, it just seems very low.
That's
And
true, but
then,
I don't know what the answer to that is. It probably depends on what neurons you're looking at.
yeah, I mean, I'm looking at these traces.
Yeah.
And it I don't know. It seems like it's a very low number for the frequency. And then my next question was, so they're doing the postsynaptic current. So they have the presynaptic neuron and they're recording from the postsynaptic cell. So the traces that we're seeing are from the postsynaptic. And then why they're holding the voltage for the inhibitory currents at -10, is that also normal for recording inhibitory currents? Or like plus 10, actually plus 10, I just realized, that's just very high to hold, a very high voltage to hold for just anything in general. So that's why I'm wondering, is that normal? -70 seems like a potential that you would want to hold the cell at to measure either way, but I'm just not sure.
What's exactly your question?
So my question for E is, why are they holding at plus 10 millivolts for the measuring the inhibitory current?
So that's just the classical way in the EFIS to record, to differentiate the iPSC versus EPSC. Those amplitudes is not exactly equal to what happens in the, let's say, physiological condition. They just somehow optimize condition to record the inhibitory versus excitatory. So the reason why they hold, so it's very good for, to spot the difference in the holding potential. -817 is close to the physiological states. And so if you should go to the method, they should use a different internal recording solution for E versus H. Those are like small details, but based on the method is valid.
Yeah. All right. So just to put everything into context, from figure 1, they saw that after prolonged activation of AGRP neurons, the daily food intake had come back to the baseline, and then I guess there's a change in body weight, but that also somehow is slightly, the effects of it is mitigated, right? So they want to figure out why this is happening. Why is it that even though you still have the hunger neurons firing, supposedly, why are you now seeing less physiological effects of those neurons firing? And one of the questions that one might have is that perhaps by prolonged exposure of CNO and activation of these receptors that they put in, maybe those neurons are no longer firing, or maybe they're not reacting to CNO anymore. But by doing electrophysiology, that doesn't seem to be the case, because they can still fire when CNO is presented. These neurons will fire much faster than in the ACSF condition. So that A shows that the receptor is not dead and CNO is still working on these neurons. So if these neurons are still affected by CNO and AGRP is still firing, why is it that they don't have any of the physiological effects, right? So that's the lingering question that's going on. But what they do also see is that it's probably some increase in inhibitory signals coming to the AGRP neurons, And that's what the electrophysiology would suggest. Okay. Right. So then figure 3.
I can take that one.
Okay.
So in figure 3, they are trying to test if there's a change in the metabolism or activity on the mice that have been activated via the CNO and the DREAD experiment. So what they do in A is just a general scheme. They're comparing all these different features they're testing, including their body composition difference, their activity difference, and later their EE, which stands for energy expenditure, and also the RER. I think that's the respiratory, what's that? Respiratory exchange ratio. So they are testing these, comparing 10 days before they start applying the clobazine and 10 days after they apply the drug. So what you can see, there's a change. That means it's like after 20 days. So so that's the scheme in A. In B, they are testing the body composition, which is lean versus fat gain or loss. So in the control one, the purple, is that purple sign? Anyway, in the control M cherry group, you can see there's just like general a little bit gain of fat because these mice are being fed. And in the blue one, in the blue group, that's the experimental group that has been weight matched to the control mice. Because remember, if the mice, they are provided with HM3DQ and they're provided with the drug, they are likely to eat more. But in order to match the weight to the control mice, this group is food restricted. Therefore, in B, in the blue dot, you can see there's a loss in the lean mass. That is a negative value. And there is also a gain, more gain in the fat mass. So that indicates there is a change of body composition between the experimental and the control group, even though the weights are still the same. Furthermore, if you look at the green dot, that is the well-fed. Oh no, that is just like they are not food restricted, so they are just more likely to take more food. So in the green bars, you only see a large increase in the fat mass, but for the lean, there's no gain or loss. So basically, this concluded that there is definitely there is a body composition change if you apply them with the DREAD activation of AGRP neurons. And in C, it's testing activity, and by measuring the beam breaks. I suppose this is a locometer test. I think they are just measuring like how frequent the mice are blocking the beam to indicate if how much the mice are traveling in the locometer. Just correct me if I was wrong.
I don't even know if it's a locometer. It might be like, you know, do you have a Nest thermometer at home?
Maybe.
You know how if you passed in front of the, you know what I'm talking about, right? These
Oh, so you just go through it and you just get it one count.
Smart thermometers that you install at home. They have these motion sensors. So that's how they figure out if somebody's at home or not.
Okay.
Yeah. So I think that's something like this where they just measure how often an animal passes a certain region in the cage.
Sure. So which indicates the activity level of the mice. Yeah. So in this case, so in the x-axis, I think the hollow or white bar indicates is the light phase. So the mice are less intended to move. And at night, the dark phase, which is there's a solid black bar, you can see they are more active. But across the three groups, I think they didn't conclude any significant difference. So that's the conclusion here for activity, no difference. And in D, you can see the three panels that are measuring the EE, the energy expenditure. And they are doing that through, well, an indirect, what they call indirect colorimetry. I think is there like a measurement, like colorimetry system? I'm not quite so familiar with this system, but it seems it's pretty computer-based indirect measurements on how much calorie the mice are spending by hour. Has anyone had any previous experience on how to measure these energy stuff? Probably not.
I thought mostly they do like respiration rates.
Yeah, they did, but the EE is also like they gain the EE measurements through through a computer program.
Yeah.
I mean the respiratory I think is relatively easy because they can analyze the air composition, right? They just use a ratio of the CO2 versus the oxygen, so they can measure the respiratory. But I don't know the energy expenditure, how they measure that. Because in method, they just say it's an indirect calorimetry. And metabolic cages operated by the energy balance core. So I think they have a facility to measure that, I believe. Anyway, so in the EE measurement, indeed, you can see that when they compared the different individual groups, they didn't see a difference among the three groups, or at least not statistically significant differences. However, in a respiratory exchange rate, they did see that if you see in the second, the blue panel and the green panel, there's a significant decrease in the respiratory exchange ratio. So that indicates there is there is an increased a lipid consumption, right? Because you're decreasing the ratio here.
Yeah, so I think these usually, if they go towards like 0.7, it means that it's more fat consumption or like fat metabolism. Then they're getting more energy through fat rather than carbohydrates. I think closer you get to 1, it's more carbohydrates. Closer you get to 0.7, it's fat.
I see. So that makes sense then, because in the control group, there is a significant difference between the control group and the other experimental group.
Yeah.
Mm-hmm. So they're gaining more fat, basically. Yeah.
Okay.
Mm-hmm.
All right, so they still haven't answered the lingering question of why the AGRP neurons are no longer having much effect in the general physiology, right? But based on maybe this effect on the RER, they thought maybe there's some change in the microbiome, which causes fat to be metabolized better, and they're getting more energy from fat. So anyone want to briefly go over figure 4? Yeah. No volunteers?
Maybe let me do it. So
yeah,
they want to look at how the gut's microbial colony changes, and then again they are having the same right now. Wait, they have another group right now. So before they have 3 groups, and right now they add dread, weight.
No, they had the pair fed in figure 1 weight match, one of the later ones, so they're using all 4.
Okay, right now they pull all the data from all 4 groups.
Yeah.
That's good, because I expect in the figure 1 I want to see like the 4 nines instead of 3.
Yeah.
That's fine. So the method is they have commercialized kits to extract the DNA from the
intestine.
Seca extract. I think it's from intestine, and what they are profiling is the ribosome RNA. So specifically for bacteria, the bacteria have 16S ribosome RNA, which the mouse neuron doesn't have. And then through this, try to see what type of bacterial species are presented in different groups. And for each column is one of those typical gut bacteria. I have no idea what they are. Some of them could be like, say, probiotics. Some of them could be like bad. So like in short term, it's good bacteria or bad bacteria for body, like say for body weight. So they virtually find no difference between each of those, across all the groups. And the B is a way to analyze, so combine all the profiling data across the group, and they use the principal. I think this is a principal analysis. So you pull out so it's basically one dot represents the feature of that group in 2 dimensions. So it's not exactly corresponding to which to a exactly bacterial species. It's probably for example, 20% of bacterial 1 and 2% of bacterial 3, let's say. I made it up. So this is like it's just a mathematical analysis. So it means all those groups somehow intermingled together. You cannot separate them. So these kind of confirm the visually conclusion from A. And then I don't exactly know what this channel diversity index is. You can just tell that there's no difference between those two, all 4 groups. And then the other thing is they try to determine how much energy. Why they need to do this? It probably relates to the energy expenditure in figure 3, I guess. Basically shows the contents or the mass of the bacteria looks similar. So you don't have in one group more bacteria than another group. So ABC probably shows the species differences. D probably shows the mass of those bacteria. They are similar. So basically meaning that so the effects of chronic activation of AGRP neurons probably, but most likely doesn't affect the gut bacteria. Mm-hmm. And it's probably from from all the figures all the experiments they show to us, the effects, so the symptom of those effects or phenotype of those effects coming from increased fat mass, but also increased fat, burning. So they gain more fat, but also burning more fat. So that's kind of interesting. That's it.
Yeah. So, well, thanks, Zhao. That's great. So throughout the paper, they've been trying to figure out why chronic AGRP neuron activation, what kind of effects it has on the metabolism of those animals, and also taking into consideration that the AGRP neurons still are they're still healthy and active, and they're still responding to CNO, but not showing the normal effects of activating the hunger neurons, right? So they're saying that there's some kind of an inhibitory signal that dampens their activity. They don't know where that's coming from. And figure 4 shows that it's maybe probably not coming from a significant change in the microbiome. So this figure 4 is really the difference between like a Nature paper and a PNAS paper. If they had seen amazing effects of AGRP activity and they saw that the microbiome had completely changed, that would be a very significant finding. But entire figure 4, all this work, all this money, effort, shows that there's no difference. So it's like a very sad ending to this paper. Right?
Yeah.
So my motivation for bringing this paper up is, of course, again, as I talked about in the beginning, AGRP neurons are the hunger neurons, and that will be where we should be looking for looking at feeding circuits, right? That'll be the most obvious target. And there's still so much unknown. If people have read the discussion, they talk about all the things that still need to be figured out. And it's a fun time to do that because we have these GQ thread system to do these kind of experiments, and very well exemplified by our activation of like the P2X2 for the taste system and interesting things that we found that way. Right? Mm-hmm. We have like 3 cages of AGRP Cree animals. So if we get around to it, we could activate AGRP neurons and see what the effects are. We could look at chronic. Here, they're just their readout is food intake. But if we were to look at which brain areas are involved in this, if we compare acute versus chronic, maybe we'll find some interesting differences in the brain regions. So I thought that I'm just throwing that out there. Just based on the findings of this paper. Yeah. Right. Anything else? Okay. We're done? Okay, let me stop the share. It's getting cold. It's getting cold out there.
Winter.